Our next panel will explore how automation can become a useful and reliable tool for biomanufacturers. Please welcome your moderator, Jeffrey Hung, PhD, Chief Executive Officer, Naujin.
Good morning, ladies and gentlemen. Welcome to the session on automation in biomanufacturing. My name is Jeff Hung, CEO of Naujin, a new startup cell and gene therapeutics company. I spent the last 10 years in biomanufacturing. First gene synthesis, then viral vector manufacturing. I witnessed and experienced firsthand the growing pain of biomanufacturing. The good news is that in cell and gene therapy manufacturing, the protocols start to be standardized, evidenced by a few large initiatives. Alliance for Regenerative Medicine published recently a gene and a cell book, respectively, to describe in detail the manufacturing process for viral vectors and cell therapies. And then Bespoke Gene Therapy Consortium, a large initiative spearheaded by Foundation for NIH, FDA, and a few industry partners, also standardized the production protocol. However, the standardization is only the beginning to automate this complex cell and gene therapy manufacturing process. Automation, even though absolutely necessary to reduce the manufacturing cost for gene and cell therapy and make gene and cell therapy affordable, automation is still far from within normalized process in the industry manufacturing. So today, we're very fortunate to put together a panel of world-class experts to explore the question such as, what it does take for the industry to adopt automation in the manufacturing process, and what the automated process look like in the cell and gene therapy manufacturing. So those are the questions this morning's panel will address. So we are very fortunate, again, to have a panel of world-class experts. So the first one present will be Eric Wu Engi, Senior Process Engineer from Takeda. And he will present on the robotics in biomanufacturing. And then Kevin Gordon, who joined me on the stage, the Chief Digital Officer at Aura Biotech, will present us a system to establish a paperless batch production records in the cell therapy manufacturing. Last but not the least, Dr. Bruce Yu, the inaugural Empower Professor at the University of Maryland and the Director of Bio and Nano Center at the University of Maryland, will present us this non-invasive analytics technology to interrogate and analyze the cell and gene therapy finished products, which is essential for automating the manufacturing process. So without further ado, let's welcome Eric, who joined us on through Zoom, and he's going to present us this robotics in biomanufacturing. Let's put our hands together.
Thank you, Jeff. My name is Eric Unagi. I'm a senior automation engineer with Takeda Pharmaceuticals, and I'm here to talk about robotics in biomanufacturing. And can we go to, do we have the slides up? Perfect. Next slide, please. So most of the content in my presentation will come from this article that was published by my colleague, David Walton. He's a member of the Bioforum group. He put together a 7-milestone roadmap to get to lights out cell therapy manufacturing in the year 2030. Next slide, please. So I'd like to start from a very high level and describe, what are the automation goals specifically for our industry. They're the sort of obvious goals of higher throughput, decreased downtime of costly equipment and using a lot of costly staff, fewer discarded batches, reducing labor costs. That can be accomplished kind of two ways. either by reducing the total number of people hours or reducing the skill set required, ideally both. But then more specific to our industry, reducing ergonomic injuries, especially in QC labs with a lot of repetitive pipetting, improving operator safety, heavy material movement, exposure to hazardous chemicals, and then also increasing accuracy and repeatability, specifically to reduce the number of deviations. There's statistics out there, it ranges from about $50,000 to $100,000 per deviation instance. So automation can, if it can help us, there's a strong financial incentive. And again, the ultimate goal here is lights out manufacturing in the year 2030. That means that there aren't human beings necessarily in the manufacturing process, and it's all robotically driven. Next slide, please. So I'd like to talk about the barriers of automation and how that relates to the industry requirements. And that double arrow there is kind of a key point. There needs to be a collaboration between us as manufacturers and our suppliers for single-use materials and consumable materials. So starting on the top left, one of the barriers is that because the cost of automation is relatively high, hardware, software, and integration, it's not really cost-effective to fully automate just looking at one processing area or one operation at a time. And so driving towards industry-wide standards and standardized solutions will allow our suppliers and vendors to use their economies of scale to reduce the cost of automation. Another barrier, compared to, for example, the semiconductor or automotive industries, those are really great examples to look at because they've been highly automated for a number of years. So a lot of that technology exists. So there are lessons that we can learn there. So, for example, a barrier on a more financial front is that total market size per repeatable manufacturing step is relatively low compared to something like semiconductors. If you imagine how many people on the planet have a cell phone, and the lifetime of cell phones is pretty short, you end up with a lot of repeatable manufacturing steps with a large market opportunity. So that drives the finances for those industries. Another barrier is that we have a high degree of vertical integration in biomanufacturing. You have raw materials, salts, powders, Wi-Fi, and cell banking coming in, and usually under one or two roofs. You'll have a bulk drug substance or some sort of finished product. And that sort of vertical integration precludes the ability for suppliers to horizontally integrate and again use their economies of scale to drive down cost. So looking at industry requirements on the right side, pharmaceutical manufacturing is very manually driven because there are a lot of sort of complex processing steps. And so we need more flexible automation systems to meet those sort of processing steps, as opposed to a sort of production line with highly repetitive steps. And another requirement is cleanability. It's not just about getting one robot in a clean room and wiping it down. It's also about being able to move materials from a not so clean space, such as a warehouse, all the way into a Class 8 area, and managing sterility as you move robotics and materials all the way through. Next slide, please. So another barrier to automation, very fundamentally, is the fact that we have a lot of bulk material movement, and it needs to be kept sterile. That's sort of unique to our industry. In semiconductor and automotive, they have bulk materials, they have cleanability, but not really both at the same time, not the way we do. And so that presents sort of fundamental challenges for the mechatronics of automation systems and for the robotics. So you have bulk material movement, again, from warehousing to dispensing to buffer prep and so on. In terms of other industry requirements, most of our facilities are brownfield facilities. They're existing buildings. And so we need sort of smaller, more flexible mechanical automation solutions that can fit into existing facilities, as opposed to having to tear down walls and put wires into concrete for position tracking and so on. Another requirement is that the processing steps have correlated documentation steps as well. It's not just about automating some mechanical operation. It's also about tracking batch IDs, sample IDs, and various metadata, and keeping in mind all of the data integrity requirements as well. Data integrity is actually a really key point for automation because a lot of software vendors in the automation space don't really have the suite of features that we need to meet the FDA data integrity requirements. So that's kind of another a strong requirement that needs to be driven down to the software vendors. And lastly, any innovation still needs to be qualified. It doesn't matter how small the step we take in terms of automating a portion of a process, it still needs to pass through validation and qualification. So that's another kind of challenge. Next slide, please. I'd like to touch on this topic of using evolution as an engineering design process. That's very common, making small iterative steps. That's how you optimize the design of a system, for example, but it's very limited in terms of making a large progress towards lights out manufacturing. So we really need to look at redesign as a process, and also driving those requirements from regulatory agencies down to our suppliers and sort of co-engineering a solution. A simple example is an automated tube welder. Tube welders, as a piece of equipment, it's designed for a human being to use it. And so it's much more difficult to get a robot, even with a vision system, to basically perform the same task as a person. So if that can be redesigned, so it's easier for automation, for robots to operate that equipment, then it's easier to integrate into your overall process. Next slide, please. Okay, so now we're getting into the roadmap itself. There's 7 milestones here. In the interest of time, I'll go through them briefly, but again, this article is available online, so you can see all 7 milestones. Next slide. So the first step in automation engineering is not to do automation engineering. It's to map out your entire process, look at your facilities, look at your materials movement, look at your processing areas, and identify which are the areas and which are the steps that are best to automate first, and integrate from there. You can have a roadmap for how do you do full integration, full automation, or just kind of focus on the most valuable areas first, and then sort of build on that. So that's the first milestone. Next slide, please. Second milestone will be to get robots to pass materials between different clean rooms, different grades. And that becomes a challenge in a few different ways. You need to build facilities that have simple things like doors that robots can actuate. You need to be able to wipe down or clean a robot, especially in an automated sort of way. And that's something that doesn't really exist today, but that's going to be necessary in order to get that fully integrated automation from materials to final product. Next slide, please. So autonomous mobile robots are a really great candidate for this. Excuse me, this is an existing technology. And the good news is that a lot of these robots actually have vision systems and LiDAR, so that they don't have to follow a predefined track. And they can sort of move around based on if there are people sort of standing in the hallway, and they can sort of move around it. So that's a good candidate for moving around materials. Next slide, please. Mobile sampling. So in terms of getting a robot to draw a sample from something like a bioreactor, aliquoting, and doing dispensing fluids into different containers, that's a solvable problem. That sort of technology already exists in terms of robotics. But the larger challenge here will be to integrate that system with LIMS, MES, distributed control systems. So getting all of that to talk together. So that's milestone number 3. Next slide, please. Milestone number 4 is to engage with suppliers. This logically follows milestone 1, so we don't have to, there's no need to wait after performing the process map. And the key point here is to engage with suppliers about the need to make robot-friendly containers. So a simple example here is if you have a tray of pipette tips that's being used in the QC lab, that's usually wrapped in some sort of plastic, in a small box, and then in a larger box filled with multiple trays. That's very difficult for a robot to open up and disassemble and to dispense individual usable, consumable materials. And so if the supplier can sort of redesign that packaging to be more robot friendly, then it's much easier for a robotic system integrator to come in and use that material in a more automated fashion. Next slide, please. Milestone number 5 is to prototype a QC laboratory with fully automated QC lab with plug-and-play mobile robots. There are some great examples from academia and also prototypes in industry that are already doing this, using things like the mobile robot with the robot arm on top to take samples, bring it to different analytical equipment within the QC lab. Typically, these will run overnight to process a lot of samples, and then during the day, staff can come in and process that data. Next slide, please. Milestone 6 is fully automated packaging facility from goods in to goods out. So, like I was mentioning before, automating a warehouse and moving material from the warehouse into weigh and dispense, into buffer prep, and so on, and being able to meet the cleanability requirements moving through those areas. Milestone number 7, lastly, is the fully automated inoculant laboratory. It seems like 2028 is a bit conservative as a milestone goal for this, but there is a decent amount of research and development that needs to be performed in order to validate that automated system. So that's the goal for inoculant laboratory. Next slide, please. And so here's a little more detail on what that can look like. There are a few different prototypes that in the industry. The Sally robot is one that's actively being developed. Next slide, please. And so lastly, that brings us to fully automated cell therapy facility. The thinking from the Bioform group is that it would be, even in 2030 with all these milestones, there would still be some staff to perform certain operations during a day shift, but then overnight, the robots can handle all the material movement and most of the manufacturing steps. Next slide, please. So those are the milestones. That's the roadmap. If there's anything you remember from this presentation, it is a call to action to think ahead of how do we design for automation. One great way to do that is to assume that there won't be human beings in the manufacturing space, assume that there's some level of automation and robots, and then work backwards to figure out how do we actually get there? What packaging do we need to redesign? What sort of processing areas or floor plan designs can we sort of redesign slightly to make it much easier to integrate automation, and collaborating with our suppliers on those requirements so that they can design solutions that work for us broadly as an industry will be very important. So not just looking at what's a highly repetitive task and how do I get a robot there, but how do I look at the overall process and integrate robots through all of it and get them to work together. The good news from a supplier side is that it's sort of a blue ocean market opportunity. This is a very unmet need in the biomanufacturing space. And so this poses a very large market opportunity for our suppliers. Next slide, please. And so I'll leave you with some actionable advice of how to, actually develop automation. So from a strategic standpoint, again, assume automation exists, work backwards. I was recently at the Boston product show for ISPE, and I heard from a few suppliers that they've thought about integrating more automation to, let's say, a fill finish machine, but there just wasn't demand from their customers. And so if we create that demand, supply will follow. More actionably, demanding standardized packaging that's robot friendly, establishing communication between your material supplier and your system integrator so the requirements can go down to the system integrator to develop the robotics that we need for things like secondary packaging. And in terms of organizations, establishing advocates internally is really important, just to socialize what technology is out there and what we need to do on the process development front in order to design for automation. Next slide. Again, the articles are online. And next slide. That's all for me today. Thank you.
Thanks, Eric. And it's a couple pieces here. One, we're trying to fit an enormous amount of content into 40 minutes, so we're running a few over. I'll try to compress 10 into about 5 or 6. Eric takes us through sort of the thoughts of, full robotics and automation for cell and gene therapy, really a reach goal. And then Bruce is going to talk to us a bit about the data side of what we're doing today. If I can get up my slides. Oh, I guess first. So hi, good morning all. I'm Kevin Gordon from Ori Biotech. Interestingly, Ori was actually founded to industrialize manufacturing for cell and gene therapy. So as soon as the slides come up, and really what I like to talk about a bit is where present state is today. Perfect. So if I can pull those up, let's see if our advancer works. So really, where is most of cell and gene therapy manufacturing today? It's in giant paper batch records, right? They're 500 pages. And then on top of that, some people put paper on glass, or a system that lives on top of those to try to digitize some of that data. But it gets really hard to get to automation robotics when there's paper transcription going on all over the facility. As a result, we get to see quite a bit of this problem for cell and gene therapy manufacturers when they go to scale to commercial, right? So a lot of failures, a lot of these you can sort of tie to tech transfer. So as people try to move from bags and flasks to more automated systems, but really probably digital adoption and their desire to move to those at the same time as changing a bunch of technology in place leads to a bunch of challenges. And then, of course, the final result of that is we have curative therapies for patients, and there's a huge population of them, and very few of them are actually receiving these life-saving and hugely transformative therapies in the market. So what can we do about it? The biggest one that we had already talked about and we sort of try to champion in this space is to really say that, you know, we need these pieces and the data out of them in really readable formats, right? So to remove paper, get things into place where they can be analyzed instead of being analog, get platforms that remove latency from the process. So if you think about Eric talking about deviations and the costs of those, a lot of times because it's a deviation that gets raised, that must go get handled manually, put back into a process, and ultimately get into place that we can actually do things like predictive analytics and multivariate data analysis and all those fun things. But for today, the first one is really just to get digital in place for these parts. So question is there a model to do this? And amazingly, there's been one since 1998, which was refined in 1995, known as the ISA 95 framework. It's machines talking to logic controllers, running through MES execution systems through ERP. Now, if you take this today and apply it to cell and gene, what you get is a picture that looks like this. It's a lot of boxes, a lot of technology to put in. And sort of as the running joke, it's like, that's simple, right? Like, it's a huge amount of pieces in place and in that stack to really start to talk to and think through. So what we at Ori have started to talk through is to go, okay, from across the spectrum of everything that's going on, from early stage research all the way through large scale automation, how can we get to simpler packages of these things that there become standard offerings in the space that start to solve all of these? And here's the fun one, is that Ori, we actually have nothing to do with the software that's gonna fix all of this. We're making sort of that same call to action to say, we need people adopting these pieces, right? So electronic black notebooks really early on, right? Remove paper as early as you can in the process. As you scale up, get to paperless batch records. The benefits of those are very quickly realized the moment that you digitize those pieces. Part of the challenge, again, as Eric talked about, is right now very few of the pieces of equipment and instruments in those facilities is really meant to talk to any of those digital systems. So it's still transcription, just removes one from paper that then tries to go digital to get directly to digital. And at the large scale, to the same concepts of cars and semiconductors, they've moved to real, so SCADA, here's one nobody will ever remember, supervisory control and data acquisition platforms. So full automation in these facilities to really start to introduce robotics on the large side. It's to work across all of these, but also to give people sort of better pre-built, pre-thought through solutions in the space. Okay, so you start to move there, what does it get you? And another one of those things that we talk about a lot, which is data's great, information's fun, but insights is really where things start to transform. And really a good example of that is, you know, to the sort of points that have been brought up, right now, a batch record for a cell and gene manufacturing process is outrageously rigid. So even if you know that in culture, you've got a pH reading that is currently trending to go above what its threshold should be, you actually can't really do anything about it in process. Well, if you can start to run those time over time to give the regulators sort of comfort in the fact that it's like, we're going to go change a variable in this manufacturing process now, you can actually start to turn that data into information to insights and start to support some of those other decisions. There's a lot of other fun that comes out of this, right? So the concept of remote or distributed manufacturing, if you can automate these processes, understand the machines that are doing them, you can do them in more than one facility at the same time. You can really start to think about what capacity you have and better understand pull, really start to think about alerting both on the biology and the mechanical side. So you think about failures that go on that cause those deviations and sort of loss of batch of a product that might be $400,000 to sell, $250,000 to manufacture. Not losing one of those is a pretty good financial incentive to keep these going forward. And everybody likes dashboards, I guess, but getting to see them visually all of a sudden does start to transform it instead of having to scan through a bunch of Excel spreadsheets. All right, outcomes, and then I'm off the stage. The biggest one that we've seen to date is the concept of release by exception. So right now, in most of these processes, you have somebody from QA/QC and technicians both touching every step of one of these processes. Once you move to digital and you put floor and ceiling values on each of those, you can actually start to say, we can just run this whole batch end to end, and as long as we don't go above or below those thresholds, we can do release in that final QA/QC testing at the end with sort of no human intervention other than the steps in the process. That improves quality. It allows for that fun of distributed manufacturing. It also means that you can use contract manufacturers as sort of buffer capacity because transferring to them gets easier as well. Your data becomes a true version of what's going on, right? Is digitally captured, becomes very immutable, hard to change in the future, gets that proactive process improvement as we do it. And then ultimately, and probably most importantly, it gets you to a lot of accuracy in location of these materials, which when you're taking cells of a person, remanufacturing them, putting them back, the one thing you can't do is cross any streams there. By having all the digital checks in and out, you can start to throw all the alarms in the world the moment one of those crosses streams. And my contact info, be happy to talk to anybody, and happy to introduce Bruce to sort of talk through the data side of all of this. Thank you.
Thank you very much. If I can have my slides, please. Hi, my name is Bruce Yu. Both Eric and Kevin alluded to the importance of data in automation, and I'm going to approach this question from the side of data collection, in particular non-invasive data collection. Here's a slide contrasting non-invasive versus invasive data collection. Biopsy, for example, is invasive, and the airport screening is non-invasive. As to how big a difference non-invasive data collection can make, just imagine airport screening involving biopsy. It's going to make the screening totally impractical. So non-invasive is important, can facilitate data collection, but that's not the only attribute we are working on. I learned a lot from the learning of the history of the evolution of computers from 1946, being a gigantic machine, to today, supercomputers, benchtop computer, desktop computers, and cell phones. So it is important for technology to be truly transformative, for the technology to be simple, affordable, and if possible, being portable as well. If you have a technology that requires a gigantic machine, a PhDs to operate, that's not going to work out that well. So the kind of technology we are developing, we call it water proton nuclear magnetic resonance, or for short WNMR. It's not the kind of NMR you typically see in the biophysics department, the biochemistry department sitting in the basement. Instead, we use benchtop instruments for product inspection and also for process monitoring. You can see the instruments are fairly compact and certainly portable, and the operation is fairly simple. And the conventional analytical technologies are invasive for product inspection, for example. You have to draw the drug substance out of the vial for data analysis, right? In the process, you destroy the integrity of the data of the drug product. Also for process monitoring, if you truly want to do inline process monitoring, currently you have to insert a probe into the processing material, and the probe gets dirty quickly, you have to clean, recalibrate, et cetera, et cetera. So if we can do non-invasive data collection, like MRI instrument scan, MRI scan in the clinic, you can monitor the patient's blood flow without the MRI scanner coming in contact with the patient's blood. So that's the idea behind our approach. So for product inspection, I showed in this picture 3 different vaccine vials, and we can put the entire vial into the instrument. You don't have to take the label off. You don't have to break the seal. Data collection takes seconds or so, 10, 15. For insulin, it takes a bit longer, a minute or so. And the advantage in water NMR, we use water as the signal source. And the advantage for that is the water is so much concentrated, right? So it's very high concentration, making data collection very, very quick. And also you have different drug products, but they use the same solvent, water. So you tweak the measurement process just slightly, you can measure vastly different products. And also you can use the flow mode instrument to measure a process. Here there's no contact, just like using the MRI machine to monitor blood flow. And here we monitor the oxidation process of propofol. You can collect data non-invasively. So they're just like multidimensional MR. We have the simpler form of WMR, where the output is a single parameter. We call it R2. Or we can make it more complex for process understanding, and maybe for product fingerprinting, we can have multivariate WMR. And there are different forms of that. I won't have the time to get into the details. So yeah, it's analogous to multidimensional MR. So we have, in collaboration with Pfizer, for example, we worked on assessing the gene capsid fading level, the capsid fading level by DNAs. And typically, I think the industry standard is you use analytical ultracentrifugation. That process may take hours, and the data collection may take hours as well. Here, using WMR, data collection takes 2 minutes per sample, and you don't have to open the vial at all. And that work has been published last year. We are currently working with industry on mRNA encapsulation inside lipid nanoparticles. And also, we applied this to vaccines. You know, manufacturing, pharmaceutical manufacturing is not very precise, and there is always difference between vials. And this difference between vials shows up here and there. In this case, it shows up in the freezing behavior. We have 5 vials of a vaccine. We subject them to freezing temperature 3 times. 1 vial didn't freeze, right? You see the vial to vial variability. But water MR can readily detect the difference. Here, vial number 9 versus the other vials. And the data collection takes about 15-20 seconds per vial. And then we use multivariate MR to differentiate 2 vaccines, very, very similar vaccines. And using different types of multivariate MR, we can gradually separate them digitally, not physically. We can tell them apart. And one potential application is counterfeit detection. These 2 vaccines are really, really very similar, but we can readily differentiate them. And also we can detect the vial-to-vial differences for very similar products. I won't have the time to get into the details, but here we have 3 products of filgrastim. Neupogen is the brand, and the other 2 are biosimilars. And we measured 3 vials of each product, and you can see they display different levels of vial-to-vial variability. And we can do that because we don't have to open the vial, and we don't have to do any sample preparation. So you can monitor or detect the subtle differences between vials. And we think this can help us to monitor the manufacturing process tightness, how tightly you control that. So in summary, water NMR can greatly help us to expand our data collection capability with the potential of 100% bioinspection before release, and then before injection if needed, and anywhere in between. And also the potential for 24/7 continuous data collection. So finally, and there's no wet lab operation at all. And finally, I want to thank my coworkers and the funding agency. Thank you.
So next we have about 5 minutes Q&A for the audience, and also we have already pre-arranged some questions just to get the discussion started. So if you want to come to the mic, feel free.
Hi, I'm Mark Shennerman from UMBC. Thank you for all the interesting talks today. Particularly interested in the last talk on NMR. Have you thought about implementing that for GMP testing, and what sort of issues would you anticipate with regard to hardware and software and validation for use in GMP?
We are working to collaborating with industry, trying to gradually adopt this technology into GMP. For now, they are exploring, industry partners are exploring the mock GMP operation. And my understanding is it's very hard to incorporate or integrate a new technology into an established manufacturing process of a marketed product. So I think what Eric mentioned earlier is important. Sort of you integrate this process, you incorporate the technology early on in the manufacturing process of a new product, like a gene therapy product or cell therapy product. But it's a slow process. We are developing the technology, and we have been collaborating with industry. We have funding from the FDA. We also had funding from Nimble. So it is an ongoing process.
Thank you. So automation, obviously, for cell and gene therapy is very complex. As an industry, I want to just ask the panelists to comment on where do you see as the weakest link as an industry, because we are as strong as our weakest link, right? So maybe start with Eric, and then Kevin, and Bruce. You are on mute, Eric.
If he is or if he's yet. There you go.
All right, can you hear me?
Yep.
Yeah.
Fantastic. Okay, yeah, so I think that's sort of a complicated question, but I would boil it down to mindset, actually. I think it's easy to look at existing operations and say, like, okay, this one step is very repetitive, so let me see if I can buy a robot off the shelf that can do that. But in order to get to more integrated, large-scale automation, you kind of need to think ahead to how do I make some maybe adjustments to my facilities or my process development to enable automation and enable robotics to come in. I think another kind of barrier is understanding the software development lifecycle. That's not something we see a lot in this industry, but understanding Scrum style project management and Agile project management and how essential that is to software development, and allocating the proper resources to allow that to happen in order to create the features to meet our regulatory requirements is also really important. Well said, Eric.
Okay.
Yeah, I think to sort of the question on GMP and to the topic of software validation, there's a big evolution that needs to happen in the life sciences industry of quality regulatory folks becoming more comfortable with these things and faster evolution in places. We, in a previous life, we digitized the manufacture of both a CAR-T and a viral vector manufacturing facility, and it really took our head of quality until we gave him a digital certificate of analysis that he knew was completely accurate to the point he got to actual comfort in these. And I think that's a company by company by company challenge that sort of needs to go through. And so it's sort of, to get to automation, it's sort of maturing an entire industry and sort of starting to pull them forward. I think really what we need is use cases of success to start to be able to better talk about and publicize.
Yeah, that's exactly right. Success speaks volume.
Yep.
Yeah. Bruce?
I don't work for in industry, but based on my conversations with industry and regulators, I think standardization would be very crucial for automation. Standardization of operation procedures and also data structures. But we collect a lot of data. How do we structure them? How do we standardize them and put them into a database as a common knowledge, like a fingerprinting of different products? I think that's a challenge from a data standpoint.
Okay. So just as a timely kind of announcement, Biden recently announced a $2 billion investment on biomanufacturing. So I just want to hear from our panelists how this initiative will help automation in biomanufacturing. Anybody? Maybe start with Kevin.
I'll take a swing at it. I think it's an interesting one in how to apply sort of those types of funding to this. I think the grants that get given and a lot of the developments that come out of them do a good job of making their way into academia. And really, I think the challenge for everyone here is to start to move and mature those as, those people from academia move into sort of the commercial side of the industry and to bring them forward. The other one is to really continue to force early into the process, right? So if you think about all the science that goes into these pieces, the thought of digital and automation as early as possible today is sort of never really top of mind. It's a lot of proof of science, which if you can do, great, because it brings in next funding round, moves you on to the next step. It's a hard sort of case to make to spend money on what people would sort of traditionally consider IT in the area, but it would be a big one if more of them adopted it early in process development and engineering run exercises, because then they learn them, get comfortable with them, and grow with them, and will sort of demand them as they move through those future stages.
Well said. Eric?
Yeah, I think Kevin had an excellent response there. From my perspective, I would just add that I think there's already a lot of market demand in biomanufacturing for automation. That's something that's sort of coming, especially for cell and gene therapy. That's I would say necessary in order to reduce the total cost to the patients. So I think any additional funding is definitely just going to accelerate that process. I remember looking through where that funding is going to be allocated, and it's going into a lot of different categories. So yeah, I'll just echo that sentiment that I'm thinking of automation and digital roadmap kind of early on, and then kind of co-engineering that so that you can plan for validating and qualifying the technology so that you're ready for it instead of kind of adopting it at the end of your design process. I think it's very important.
Okay. Bruce, do you want to add anything?
Yeah, just very quickly, I think the promise of automation is higher quality and lower cost. And these two features, I think, would be crucial to bring manufacturing back in this country, right? To have higher quality, but at a lower cost.
That's a good remark and the final goal for this automation in biomanufacturing. So due to the time constraint, let's put our hands together to thank our panelists.
Thank you all. Thank you. Right.